Okay, let's take a look at 16.6. Again, you should go take a look at problem 16.5 because it's a nice lead-in to this problem. So I'll quickly sketch the lac operon again. So we got lac I here, promoter, operator with the lac I binding site, lac Z, lac Y, lac A, we call this lac O, this is the promoter. Okay, so let me copy this so I don't have to keep drawing that. I'm going to store that down here for now. Okay, so our first question here is, okay, well, what is the question for 16.6? For the genotypes and conditions, lactose present or absent shown in the following table, predict whether functional enzymes, non-functional enzymes, or no enzymes are made. Sorry, did I say or made? Okay, so our first one is done for us, pretty much. Okay, lac I plus, lac O plus, lac Z plus. So everything is plus here, plus, plus, plus. Okay, great. Essentially, this is the normal lac operon. So we know that when lactose is added to this, what's going to happen? Well, here's the no lactose situation. Okay, what happens when no lactose is made? Now remember, lac I is always expressed and it's regulated differently from the lac operon. So it's expressed constitutively. It's always on, and so it's going to look like this. And when lactose is not around, the lactose binding site will be free, and as a result it will be bound and it will prevent transcription of the structural genes. Okay, right? So RNA polymerase cannot transcribe when lac I is bound, and lac I is bound when there's no lactose around. Okay, so that is why we have a plus or whatever X under no enzymes made when there is no lactose. Okay. Now, okay, we have our next one. We have this genotype right here. So very similar, except we are going to replace the wild type operator with the constitutive mutation. And what is that? Well, that is, there's my eraser here. That is this thing right here. The binding site has been mutated. Okay, so the lac repressor cannot bind So, and the question is telling us, okay, well, lactose is around. Okay, so lactose is around. So lactose is going to be bound to the repressor, and the repressor is not going to be, it's going to have that allosteric shift anyways, so it's not going to bind anyways. It couldn't bind if it wanted to because this is the constitutive mutation. So lactose is around. We have the OC mutation. RNA polymerase is going to transcribe all of those. So I'm going to put functional enzyme made. Right? There's nothing to block. There's nothing to block the polymerase here. Doesn't matter whether lactose is around or not because we have the OC mutation here. So functional enzyme will be made. Okay. So let's go on to the next one. So for the next one, we have trying to change the color of my pen. Okay, for the next one we have LAC-I minus. Okay, let's change that. Okay, so LAC-I minus, let's go with pink. LAC-I minus, O plus, okay. And Z minus, okay. I'm just going to cross that out like that, put Z minus. Okay, so this is the genotype of the E. coli cell, and we have no lactose. Okay, so no lactose is here. So what's going to happen? Well, LAC-I minus, I should erase this too, right? So we have the LAC-I minus mutation, which means this site right here is mutated, so it can never bind the operator. The operator is the wild type, so we have the LAC-I repressor binding site, but because this is the LAC-I mutation, it doesn't matter, it's never going to bind here. So no lactose. Okay, so this is the lactose binding site is unoccupied. It doesn't matter whether there's lactose there or not. This can never bind the operator. Okay, so what's going to happen? Well, RNA polymerase is going to come in here and it's going to try to transcribe these genes, and it's going to be very happy because it can. It's not blocked. Okay, great. But what it's going to do is going to make, it's going to transcribe this long RNA with open reading frame, the codons for LAC-Z, Y, and A, but what's wrong with Z? Z is mutated. We have Z minus here, so Z is going to be non-functional. So, after all that, we have non-functional enzyme made. Okay, the enzyme's going to be made, but it's non-functional. Okay. Let's take a look at the next one. The next one is, okay, good. I like when they do this. It's the same genotype, but now we're looking at this genotype in the presence of lactose. Okay, so let's take a look here. Lactose is now going to bind this mutated LAC-I. The lactose binding site is okay, so it's going to bind here. But oh, nothing's going to happen because, you know, this is the LAC-I mutant, and the operator binding site was, or binding residues, the part of the protein which actually bound the operator was mutated to begin with. So it didn't matter whether lactose was bound to the protein or not. It doesn't matter. So it can't bind there. So same thing's going to happen. This can't block here. RNA polymerase is happy. It's going to transcribe all the structural genes, but Z is still mutated. So we have a non-functional enzyme made under these conditions. Okay, so let's take a look at the next one. It has a mutated operator binding site. It can still bind lactose. It doesn't matter though. It's not going to, the allosteric shift induced is going to do nothing. It still cannot bind the operator. This one right here, however, you know, is perfectly functional. It can bind lactose, and lactose will induce the shift up here. Okay, so in this cell, when there is no lactose, what is going to happen? Okay, well, this cannot bind. So this cannot bind the operator. So this is useless to us. It's just going to be floating around in the cell doing nothing. But this one over here that's encoded from the plasmid, well, it can float around in the cell, and it can still bind to the operator up here. Because it can bind to the operator when there's no lactose around, right? So if there's no lactose, it can bind to the operator, and it will block RNA polymerase. And because RNA polymerase is blocked, no transcription of the structural genes, and no enzyme will be made. Okay. No enzyme made. Let's take a look at the next one now. So these are long problems, aren't they? I hope they're making sense. If you're having trouble and you're trying to watch the video, I hope they're making sense to you. One of the drawbacks to these videos is you can't stop me and ask me to clarify something. So I can only hope that they're offering some help to those of you who couldn't make it to office hours or review sessions or something like that. Okay, the next strain. Let's take a look here. Okay, so I wish I had a bigger eraser here. Okay, I got it. Copy this and just cut it. Okay, that works. I'm going to cut all this too, and I'm going to copy this. Put that here. Okay. Now, okay, for this one, let's see, we have on the normal chromosome, I think I need to get a new pen here, and on the plasmid. Okay, so on the chromosome, we have normal LAC-I, we have mutated LAC-O, the constitutive mutation, so the LAC-I binding site is mutated, and we have normal LAC-Z, okay? And on the plasmid, we have a normal copy of the operator. Okay, so what does this mean? So let's go up to our model of the LAC operon. Okay, so everything, this is a plus up here. This is the constitutive mutation, so I'm going to need to erase the binding site for the repressor. And all these other genes are plus. Okay, promoter. We'll say the promoter is normal too. Okay, so in the presence of lactose, what is going to happen? So LAC-I is making the LAC-I repressor. It's encoding the messenger RNA. It's being translated into the LAC-I repressor. Lactose is coming over here. It's binding to the repressor. It's causing the allosteric shift. So I'm going to actually draw that. Okay, and okay, so now it cannot bind to the operator, and it can't bind here anyways, right? Because this one right here was the constitutive mutation with the binding site mutated. However, on the plasmid, right? This is the plasmid. There's a normal copy of LAC-O plus. Okay, so over on the plasmid, this guy, if lactose wasn't present, it could bind there. But lactose is present, it cannot bind there. So this is just going to be floating around in the cell. Even if it could bind there, nothing would happen. Why? Because RNA polymerase is going to bind to the promoter, and it's going to transcribe all of the structural genes, whether or not lactose is in the cell, because we have the OC mutation here. So the repressor cannot bind whether lactose is there or not. Okay, so this means we are going to have functional enzyme made for this one. Okay, let's take a look at the next one here. Okay, so the next one is a little confusing, and I'll explain why. So we're working on this one right here, and it's got a long genotype because we have a bunch of genes on the chromosome at the normal location for the LAC operon, and we also have a bunch of different genes on the plasmid too. So I'm going to take this, I'm going to put the spare one up here for now, and then I'm going to cut that out. And I'm going to put this right here. Okay. So this one right here, okay, this is going to be our copy of the operon on the chromosome. Now, and we have a wild-type copy of LAC-I, wild-type copy of LAC-O with the normal LAC-I binding site, and mutant copy of LAC-Z. And then we can say plus for the others. Doesn't really matter. We're mostly interested in LAC-Z. So, okay, so that is what the LAC operon looks like on the chromosome. So what about the plasmid? Okay, so normally we just, when we're looking at the plasmid, we're saying, okay, well, we have a copy of O, or we have a copy of I. In this case, we're going to have to say we have a copy of LAC-I. Okay, so LAC-I plus. And then we have, and maybe it's not quite connected, just like in the normal case, maybe it's not right next to the other genes, but it's still on the plasmid. So we're going to have our promoter here. See, it's not indicated in the question, that's why it's a little confusing. So we're going to have a normal copy of O, and then we have a normal copy of Z. Okay, so this is the plasmid over here. This is the chromosome over here. And the question is telling us lactose is present. Okay, so let's diagram the lactose. Lactose is present everywhere. And I have this line here, but keep in mind that these aren't really separated in the cell, right? They're on different DNA molecules, but they're in the same cell. Okay, so both LAC-I copies of the LAC-I repressor are going to make, normal LAC-I repressor. Okay, they're both transcribed, whether lactose is there or not, and they make normal LAC-I repressor with the operator binding site and the lactose binding site. Okay, and both copies of the operator are normal. So lactose is going to bind to both of these and cause the allosteric shift in both of them. So neither of them are going to be able to bind to either LAC-O operator. Okay, great. So we're going to have RNA polymerase come in and bind to the promoter, and transcribe a non-functional copy of Z. Okay, so here's our messenger RNA with Z, Y, and A. Z does not work, so we get a non-functional Z protein being translated. But RNA polymerase is also going to come in here, bind to this promoter. You see, we have to know the promoter is there. If the promoter is not there, then you can't transcribe the genes anyway. So the promoter is there. The question didn't indicate it, but I took a look at the study guide, and apparently the authors want us to consider that the promoter is there because this is transcribed. So this RNA will be transcribed, and the problem also didn't say if Y was here or A was here, but we can ignore those. Z is going to be transcribed. It is functional. So this is the messenger RNA. Functional Z will be transcribed from the plasmid. So in this case, functional enzyme will be made and non-functional enzyme will be made, right? Functional enzyme from here and non-functional from over here. Okay, so for the next one, they changed it up on us in two ways. What did they do? They took away the lactose They're making things difficult on us. I wish they would just do one change at a time. Okay, they took away the lactose and they mutated one of the repressors. Okay, let's see how we can change this diagram. So no lactose. So I wonder, maybe I should cut some things. I'll cut some things out. That will be easier. So I'm going to let's cut all of that. Let's cut that. And let's cut that. And I think we're okay. Cut that. Okay, now let me fix some of the things here. So we have mutant copy of lac-I over here. Okay, and we have no lactose in the cell. Okay, so lac-I minus, we've already determined that lac-I minus is always mutated in the operator binding site. Okay, so this is our lac-I minus protein. This one over here, this is still lac-I plus. Okay, so this one is still free to bind to lactose and undergo allosteric shift. There's no lactose present. Okay, so this is going to be bound to this one over here, this operator on the plasmid, and it's also going to be bound over here, right? It's free to move around the cell, binding to any operator with a non-mutated lac-I binding site. Because it's not just specific for the plasmid, it can go to the chromosome too. Anywhere there's a lac-I binding site, it's going to bind. There's no lactose, so it's going to be bound. This doesn't matter. The mutated lac-I, it can't bind to either one because the operator binding site is mutated. However, this one is always going to be bound, and as a result, the polymerase is going to be blocked in both cases. So this one will not be transcribed, and this one will not be transcribed, so neither will be made. So no enzyme made for this one. So that wasn't too bad. They changed the strain on us in two ways, but no, they changed it in one way, but then they took away lactose, but not too difficult. Okay, so we have two more strains to look at. I think, what will I do? Let me quickly cut this stuff away here. Okay, and re-diagram some of it. Okay, so on the chromosome, we have the suppressor version of lac-I now. Then we have promoter, normal operator, and normal copy of lac-Z. And I can put Y in here, and A. Now on the plasmid, we have just a copy of the operator. Okay. So on both the chromosome and the plasmid, the operator is normal. And the only funny thing we're looking at here is the super suppressor mutant of lac-I. So what does this look like? So this one has a functional operator binding site, but a mutated lactose binding site. So this is always going to be bound to the operator. It doesn't matter whether lactose is there or not, it's always going to be bound. I didn't even look at the question yet to see if the authors are asking or telling us to consider the case when lactose is present or not present, because it doesn't matter. The super repressor is always going to be bound to both operators on the plasmid in the chromosome, whether lactose is there or not. And the authors say no lactose, and I say, who cares? I say, who cares to that? So none of these enzymes are going to be made. Okay, just like that. That was easy. Okay, for the last one here, let's take a look. Okay, for the last one here, hope it's an easy one. Let us cut this stuff away again, because I think that is easiest for me. It takes a bit longer, but it is easiest, I think. So diagramming this operon again. Remember, the lac I is not part of the operon, so I'm indicating that with a little hash mark here. So hash mark or hatch mark? I'm not really sure how to describe that. Lac I plus. Okay, good. Now let's diagram the operon. So P, and we have OC. Again, I'm getting a little tired of OC. Z plus, Y plus, and A plus. And on the plasmid, we have O plus and Z plus. And I'm going to put P in here, the promoter too. So let's put the binding sites in for the repressor, only over here, not over here, because this is the constitutive mutation where the DNA sequence is mutated. DNA sequence for lac I binding is mutated. Okay, so this is making a repressor, normal repressor. No repressor from the plasmid. Okay, great. Lactose is present. So let's put our lactose molecule in the lactose binding site, and then let us remove the operator binding site. Lactose binding causes the allosteric shift. Removing the operator binding site. Okay, so this cannot bind here, cannot bind there, and I think this is too easy. So RNA polymerase is going to come in and it is going to transcribe all of these, and it's going to transcribe this one here. So functional enzyme made. Okay, I hope I didn't make any mistakes there. I don't think I did, and we can go on to the next one now, which will be 16.7.